Hello students, welcome to CBSE NCERT Solutions by Vedantu. I am Miss Gopika, your own biology master teacher. So good morning all of you. Today is a very early class. So today I'm going to do sexual reproduction in flowering plants, and yesterday I had done a small part of it which I could not complete. So today I thought I will continue from where I taught yesterday. So before I continue with the class, let me also see if we are live so that I can start the class smoothly. Yes, we are live. Yes, we are live. Very nice. Okay. So yesterday's part, I don't know how many of you saw. It was a premiere video, so all of you can go and check sexual reproduction and flowering plants. I have given the introduction to what is flowering plants and how it is useful for the humans. And today and yesterday I also did microsporogenesis. Okay, how the pollen grains are formed. So pollen grain is the male gamete and ovules are the female gametes, right? This two has to fertilize to have fertilization event to happen, and then the seed formation and etc. So the pre-fertilization events were preparing these people, telling that okay, wait, you are going to get married, like how we get married, no, before the marriage, what and all things that we do, right? Be it the bride getting ready, be it the groom getting ready, be them seeing each other. So like that, pre-fertilization events are there, which is preparing this pollen grains formation, pollen tube germination, etc, etc. Now fertilization stage will come, and again post fertilization stage will come. So we are going to learn that. So students, this is how an anther look, and this is one of the very important concepts of your chapter as well. Okay, important concepts of your chapter as well. Anther formation. Okay, anther, and this anther is very you have to remember a lot of things about this anther. That is, this is dithecus. I've explained in yesterday's class about dithecus. Okay, I have also explained dithecus means in each lobe theca will be there, right? This is how the lobe is. Okay, this is how the lobe is. In each lobe, two thecas will be there. So we will call it dithecus. And to keep these two lobe together, we have a connective. Okay, we have a connective. And we can tell, first important thing is it is bilobed. Okay, we can tell tetrasporangiate because each of the microsporangia, 4 microsporangia is there. So we can tell tetrasporangiate. Okay, now students, in between this microsporangia, in the center region, there is going to be a set of compact cells. Okay, a set of compact cells called as porogenous tissues. I want all of you to mark your attendance by liking the video and also considering to subscribe the channel as well, because this channel gives you a board preparation. Okay, board preparation. Voice is also clear? Yes. Cells. Voice is also clear. Okay, all of you can mark your attendance by liking the video. So sporogenous tissue. Now this sporogenous tissue, like I told you yesterday, okay, here then we saw about the different types of layers of it. Now as I told you, this sporogenous tissue, let's take this. Okay, sporogenous tissue. Okay, so this tissue can form the MMC, that is the microspore mother cell. Okay, microspore mother cell. Okay, microspore mother cell it can form. This microspore mother cell will form microspore tetrad. This is the tetrad. Tetrad means 4. Okay, tetrad dot. Okay, this tetrad. But there is a problem. The issue that I told you yesterday was that there is a callose. Okay, callose layer coating this tetrads. What is that? A callose layer. Okay, callose layer that is coating this microspore tetrad. Now imagine if 4 of us are there and all 4 of us are stuck by something. Can each of us show our potential? Can each of us go into different fields of life? Can each of us, even do something? We cannot do something, right? So same way, even this microspore tetrads also, they are kept together. If they're kept together, then there is no way that we can go somewhere, or this microspore can form the pollen grains. So that is when tapetum will come into help. Okay, that is when tapetum is going to come into help, and tapetum will not only nourish the forming pollen grains, okay, but it will also release or secrete, okay, release or secrete callase. Okay, callase. This callase will go and break this callose, and like that this microspore tetrads, okay, this one will form 4 separate microspore tetrads. Okay, 4 of them separate microspore tetrads will be formed. Now each of them has the ability to form a potential pollen grain. Okay, this is a pollen grain. Yes, very nice. Okay, okay, here this is one pollen grain. Let's say this is the X sign. Okay, let's leave the germ pore. Okay, X sign, vegetative cell, generator cell. Each of them, okay, each of them can become a pollen grain by itself. Each of them have the potential to become a pollen grain. You will understand why I am using the word potential when I speak about megasporogenesis. So this is also pollen grain. So each of them will have the ability to form a pollen grain. Okay. So the sporogenous tissue, you shouldn't forget, students, here sometimes they also use pollen mother cell. Okay, PMC, pollen mother cell they'll use. You can use microspore mother cell, because same thing will go in again megaspore. Okay, megaspore mother cell, again they'll form microspore tetrad. And each of them will become a pollen grain. Now let's move forward. So I've done a slight revision of what I did yesterday. Now look here, microsporogenesis. As the anther develops, the cells of the sporogenous tissue undergo meiotic division to form microspore tetrads. That's why I told you, you can neither write sporogenous tissue, okay, you can neither write sporogenous tissue, or you can write MMC. Okay, anyone you can write, or you can write sporogenous tissue, MMC. Okay, matures to form MMC, that is with the help of meiosis, then forms microspore tetrad. Microspore tetrad. Got it? And then this microspore tetrads will form pollen grains. Pollen grains. Okay, clear? Now, this one. As the cells of the sporogenous tissue is capable of giving rise to a microspore tetrad, each one is a potential pollen or microspore mother cell. This is what I told you, okay? The process of formation of microspore from the pollen mother cell through meiosis is called as microsporogenesis. So here instead of MMC, you can use PMC as well, and the process of forming microspores, forming microspores from this is called as microsporogenesis. Okay, yes, microsporogenesis. Sporogenesis. Here meiosis is happening. Okay, meiosis is happening. Clear? Okay, fine. Now the microspores, as they are formed, are arranged in clusters of 4 cells. That is called a microspore tetrad, which I already told you. As the anther mature and dehydrate, okay, as the anther mature and dehydrate, the microspore dissociates. Okay, the microspore dissociates from each other and develop into pollen grains. Inside each microsporangium, several thousands of microspores of the pollen grains are formed that are released with dehiscence of the anther. So students, so much is being formed, right? So much is getting formed. Now all of that has to be, see, this is an anther. Yes or no? This is an anther. Now inside this, so many are formed. So many pollen grains are formed. What is going to happen? they are going to dehiscent. Okay, they are going to dehiscent. Obviously they are not going to dehiscent like this. Okay, they will dehiscent. That means there will be a line of dehiscence. Okay, there will be a line of dehiscence, and let's consider this OD to be the line of dehiscence. Okay, like this. So this line of dehiscence, this dehiscence is also helped by who? Helped by your three people. Okay, that is your, don't forget, epidermis, endothelium, okay, and your middle layer. Okay, three people are helping in this. Clear? Okay, chalo. So these three people are going to help in the line of dehiscence. Otherwise, forming the pollen grains, what if you form the pollen grains and there is no line of dehiscence, anther is not even ready to dehiscent, then how it is going to further? It will not happen further, no? So line of dehiscence is very important. Even though they are doing protection and everything, line of dehiscence also is very, very important. Now look here, the pollen grain. See, now I have dust allergy. Like that, some of you might have pollen grain allergy. Okay, I for me, as soon as I wake up in the morning itself, for me immediately sneezing starts happening. That is because I have slight allergy when the fan is on, or if there is any dust in the room. Okay, I will immediately get allergy. Now it is not possible to clear all the dust. Same way goes with pollen. Okay, so now you are giving flower to someone you love and that person have pollen grain allergy, your entire relationship will be screwed. No, same thing happens. That is what, like, in a sense, you have to know if that other person have pollen allergy. Some people know like maybe just casually you're going to give the flower, immediately they'll start sneezing because pollen will get scattered immediately. Right, so pollen grains represent the male gametophyte. Now students, this is very important because people get confused with gametophyte. So pollen grain is a male gametophyte. So here you can ask me a question, ma'am, then what is a gamete? Okay, wait, we will see. If touch the open anther of hibiscus or any other flower, you find a deposition of yellowish powdery pollen grains on your finger. Sprinkle these grains on water and take it under the microscope. Okay, each of us do not have a microscope at home, so we cannot do that. But I think all of you would have seen, no, in a hibiscus plant, if I like, let's say this is the stamen, I mean this is the filament, all of you would have seen entire anther will be covered like this by powdery, powdery yellow color thing, and when you touch it, you immediately feel that in your hand and you'll be able to exactly spot it as well. Yes or no? This is how it will look in hibiscus, right? So these yellow color things that you're seeing are pollen grains. Okay, pollen grains. Yes or no? Now let's go further. You will be really amazed at the variety of shape, size, colors that and designs that are seen on earth, like the pollen grains. So each of it, if you see it under microscope, we will be amazed about the beauty of it. Now pollen grains are generally spherical, measuring about 25 to 50 micrometer in diameter. So pollen grains are generally spherical, measuring 25 to 50 micrometer in diameter. It has two prominent layers. So now if I have to draw this, there is no shapes available here, students, otherwise I would have drawn it with shape. Okay, spherical. Okay, this is the thing. Now they are telling that pollen grains have two layers. Okay, pollen grains have two layers. So I'm going to draw the first layer, that is going to be the outermost layer. Okay, outermost layer. This is a very diagrammatic representation. Okay, this is the outermost layer, and the inner layer is going to be obviously this layer. Okay, this is the inner layer. Does it look bad? Sorry students, I'll draw it again better. Okay, this is a pollen grain. Let's draw it smaller. This is a pollen grain. Even this is going to be a pollen grain. Okay, and they are telling pollen grains have two outer layers. Okay, two outer layers. So this is the outer layer, exine. Okay. Okay, same thing goes here. So here, this layer which I've drawn here is called the exine layer. Okay, and this exine, students, listen carefully, it is made up of sporopollenin. Okay, it is made up of what? Sporopollenin. Very, very important. Okay, very, very important. Are you all following me for this? Sporopollenin. Okay, NIN. In spelling I wrote this, not visible. Sporopollen. Now, ma'am, what is so special about the sporopollen? You can see what I'm writing. No, yeah. What is so special about the sporopollen? Let me tell you that in right now, the sporopollen is one of the most resistant organic material to ever exist. Okay, organic material to ever exist. Why? Because acid, they have tested acid on it, they have tested high temperatures on it, but strong acids, temperature, everything has been tried, but the sporopollen hasn't moved an inch. That is, students, sporopollen is so, strong and resistant that there is no material found yet to destroy the sporopollen, and that could be one of the sole reasons why the pollen grains are able to reach at a distance and they are able to germinate in a different place, right? They're able to do fertilization is because of this resistant layer. Now, students, inside, okay, the next layer, which is continuous. Now, if you look here, I have given spaces. So this exine is not a complete layer. Okay, it's an incomplete layer. It's an incomplete layer. Now, why am I telling incomplete? Because can you see spaces I have left here? I have left it with knowingly. That forms the germ pore. So it's because this is very resistant and a very hard layer. How will the pollen tube germination happen? If pollen tube germination has to happen, then there should be a space, right? Otherwise the pollen tube germination will not happen. So the space that is given here, okay, the space that is given here is called the germ pore. Okay, now the next layer, the inner layer, okay, this is the inner layer. The inner layer is going to be called the intine. And intine is a continuous layer. You can see it is a complete continuous layer that I've drawn. Okay, now let's read what they're telling in NCERT. It has a prominent two-layered wall. The hard outer layer is called exine and it is made up of sporopollen, which is one of the most resistant organic material known. It can withstand high temperatures, strong acids, and even alkali. No enzyme, this is also very shocking, right? No enzyme that degrades sporopollen is so far known. So that means sporopollen is like the top, okay, the top of everything. Now, pollen grain exine has a prominent aperture called the germ pore. Okay, aperture means a small opening called the germ pore where the sporopollen will be absent. For this will allow the germination of the pollen tube. Otherwise, pollen tube germination will not happen. Okay. Pollen grains are well preserved as fossil because of the presence of sporopollen. The exine exhibits a fascinating array of patterns and designs. See, now I have drawn like this. It can also be different, right? It can also be different. Let's say if this is a pollen. Okay. They can also have different kinds of pattern. Can have any kinds of pattern. Okay, so the sporopollen exists or show a different kind of pattern. Got it? Exhibit a fascinating array of patterns. See, what do you think that, why do you think that exine should be so hard? Obviously, for the main or one sole reason that the pollen grains will get transferred with the help of agents, right? Agents are going to do it. So for pollination to happen, we need a pollen grain that will go and safely go and land on the stigma. So this pollen grain has to have a hard resistance, so it can be, you know, not damaged during the transport. Yes or no? No transport. While any transport is happening, no damages will happen. Okay. I'm so sorry, students. Give me 1 minute. Yes. Okay. So that was clear. Yes. Now, the inner wall of the pollen grain is called intine, and it is a thin, continuous layer made up of cellulose and pectin. Very, very important. They are telling that intine, okay, intine is the innermost layer. Innermost layer, and this innermost layer is made up of, okay, this innermost layer is made up of cellulose and pectin. Cellulose and pectin. Okay. Now, the cytoplasm of the pollen grain is surrounded by a plasma membrane. When the pollen grain is mature into two cells, it will have a vegetative cell and it will also have a generative cell. Okay, it will have a vegetative cell and it will have a generative cell. Okay, awesome. So here, look here, when if I draw a pollen grain, okay, if I draw a pollen grain, they are telling this cell will have two regions. Okay, two regions. They'll have a plasma membrane and they'll have two regions, one region here and one region here. So the larger cell is called the vegetative cell. Vegetative cell. The smaller cell is called the generative cell. Generative cell. Let me tell you that vegetative cell provides the nourishment for the generative cell. So inside the generative cell, you are going to find your male gamete. Because pollen grain is a gametophyte. You cannot tell that pollen grain is become. Okay. Hi Vishal. Yes, everyone is here. We cannot tell that, you know, pollen grain is like, definitely pollen grain is the male gametophyte, no doubt in that. Male gamete is going to be formed from the pollen grains. The male gamete that will go and fuse with the egg is going to be present in the generative cell. Okay, so here you can see the vegetative cell is bigger, has abundant, okay, food reserve. Bigger, has abundant food reserve and a large irregular shaped nucleus. Okay, the generative cell is small and floats in the cytoplasm of the vegetative cell. It is spindle shaped with dense cytoplasm and a nucleus. Okay, so both of them have a bigger, like in the sense, vegetative cell has a bigger and an abundant cell because obviously they have to do the food reserve. Now students, your storeroom will have more collection than that is in your kitchen. No, that is why, because you have to take from the storeroom. Right, every month when something, the ration gets over, what you will do, you will go to the storeroom and you take and come. So storeroom will have more content. So same way, vegetative cell has to provide nourishment for the generative cell. So obviously vegetative cell will have more reserve material, right? More reserve material. So look here, cytoplasm of the vegetative cell. It is a spindle shaped with dense cytoplasm and a nucleus. This is what is spoken about your generative cell. Okay, that they are spindle shaped and dense cytoplasm. Okay, dense cytoplasm. Now in over 60%, very important sentence, in over 60% of the angiosperm, pollen grains are shed at two-celled stage. What is two-celled stage? That is one vegetative stage, one generative cell. This is two-cell stage. Okay, two-cell stage. But in the rest of them, okay, in the rest, so this is happening in 60% of the angiosperm. In two-cell stage, this will dehiscent. Okay, the remaining will dehiscent, but the rest of the 40%, what is going to happen? 3 cell stage will be there, where there will be 1 vegetative cell, okay, plus 2 generative cells. That will be forming the 3 cell stage. 3 celled stage. So the 40% of the dehiscence, okay, 40% of the dehiscence will happen in the 3 cell stage. Okay, got it? Yes or no? Got it? So 60%, so 1 vegetative cell and 1 generative cell will form 2 celled stage. Okay, 40% means 1 vegetative cell, 2 generative cell will form the 3 cell stage. Okay, 3 cell stage. Now students, we'll just write what we wrote. So in the middle I'll have blank slides. In the blank slides I'll just give you an idea about what you should do. Okay, got it? 1 second. Yes, ready? Yes, pollen grains. These pollen grains are going to be, most importantly, male gametophyte. Okay, male gametophyte. Okay, this male gametophyte will have the pollen grain. So the pollen grain, this is the pollen grain. On the pollen grain, 2 layers are going to be there. This is the outer layer called the exine. Okay, and then there are spaces. Yes or no? There's going to be a middle layer. It is going to have an irregular nucleus and a spindle shaped nucleus. Okay, now obviously inside the cytoplasm, okay, other organelles, everything will be there. So now look here, what is going to happen here? Okay, this one, this outer layer is exine, okay, which is made up of sporopollen. Sporopollen. Okay, then this inner layer, okay, inner layer is called intine, which is made up of cellulose and pectin. Cellulose and pectin. Okay, hi Paul. So students, all of you mark your attendance as well. Okay, come on, all of you should do that. Yes, look here, eccrine is sporopollen, intine is made up of cellulose and pectin. Okay, cellulose and pectin. Now students, here there are spaces or apertures. These apertures are called germ pore. Okay, germ pore. Germ pore is through which the pollen tube will come. Now the larger cell is called as the vegetative cell. Vegetative cell. And the smaller cell is called as the generative cell. Generative cell. Got it? Vegetative cell and generative cell. Now here exine, cellulose, okay, understood ma'am. Generative cell is small, spindle shaped, okay, and vegetative cell is a food reserve. Okay, it's a food reserve and it is going to provide all the nourishment. Okay, food reserve, and it's going to provide all the reserved, whatever reserved material they have, all of it they are going to give it to the generative cell. Okay, and inside the generative cell you will have the male gamete. So it's a very important cell, right? It will have the male gamete. Now students, 60%, very important. Okay, 60% angiosperms will dehiscence. at 2 cell stage. What is 2 cell stage? One vegetative cell, one generative cell. Okay. Now the rest 40%, this is again a very like direct approximate, like that we tell no value. They dehiscence at 3 cell stage. That is one vegetative cell plus 2 generative cells. Okay, 3 of them will dehiscence at the 3 cell stage. So this is important. Okay, I hope it is clear, visible, right? Yes, perfectly visible. Clear, visible? Perfect. Let's continue. Now, pollen grains of many species causes severe allergies and bronchial affiliation. Bronchial affiliation is the swelling in the bronchus, because of which that will lead to respiratory disorders. It can also cause asthma, bronchitis, etc. It may be mentioned that Parthenium or a carrot grass that came into India as a contaminant with imported wheat has become a ubiquitous in occurrence and causes pollen allergy. I don't know how many of you have seen this, carrot grass. It will be present everywhere, wherever the plants are not there or weeds are there. No, you will be able to see carrot grass, which is like this. Like this it will look, okay? They'll have a white color, small, tiny flower, which is also called as Parthenium. Parthenium or carrot grass. Okay. I have dust allergy. You people, some of you might have pollen allergy, right? Some of you might have pollen allergy. So yeah, this is the symptom of my dust allergy. Okay, I keep having it oftenly. Yes, pollen grains are rich in nutrients. Okay, they are rich in nutrients. It has become a fashion in recent years to use pollen tablet as a food supplement. In Western countries, a number of pollen products in form of tablets and syrups are available in the market. Pollen consumption has been claimed to increase the performance of athletes and race horses. Okay, so some people actually have or consume the pollen tablets as a supplement in Western countries, not very common in India. Okay, supplements. Okay, now let's talk about how they will be preserved. Okay, preserved, etc. like that. See, once they are shed, pollen grains have to land on the stigma before they lose their viability. That is, viability is the ability to fertilize, right? If they have to bring about fertilization, how long do you think the pollen grains retain viability? The period for which the pollen grain will remain viable is highly variable for different species to species. It is going to be differently variable. Okay, highly variable, and to some extent depends on the prevailing temperature and humidity. Yes, that also definitely matters. So how can we tell that, okay, you will be staying for 1 hour, viability will be for 1 hour for rice? You cannot tell that, right? So that means what? Every plant has its own viability, or every species has its own viability. Okay, this period is completely different. Okay, it's completely different. Why? Because it depends on 2 other factors as well, like humidity and temperature. Humidity and temperature. Okay, so here prevailing temperature and humidity. In some cereals such as rice and wheat, pollen grains loses viability within 30 minutes of their release, and in some members of Rosaceae, Leguminaceae, Solanaceae, they maintain the viability for months. Okay, so this is the way to remember this. Okay, look here, rice and wheat seeds will be viable for 30 minutes. Okay, viability is for 30 minutes. Now roleso or rosore. Okay, rosore. I was just seeing how you can remember this. Rosole. You can remember like this also. Rosole, that is your Rosaceae family, Leguminaceae family, Solanaceae family. They can maintain viability. Okay, they can maintain viability for months. Okay, for longer months they'll be able to, but rice and wheat within 30 minutes. Okay, so they are maintained viability for month. You may have heard, okay, you may have heard of the storing semen. Okay, storing semen, sperms of many animals. Okay, so storing semen of and sperms of many animals including humans for artificial insemination. It is possible to store pollen grains of a large number of species for years in liquid nitrogen. So students, if you want to do preservation, right, like nowadays people also do no preservation, that at the age of like 30 they are not married. What they do, they take the sperm and ovum and they preserve it, telling that okay, if at 40 if I want to have my own child, everything is going to be safe. Same way we can also store the pollen grain, so that depending on our need, we will be able to do the viability. Okay, we'll be able to do the viability. So this is called as cryopreservation. Cryopreservation, where you will be able to see that the pollen grains can be preserved or stored in liquid nitrogen. Okay, liquid nitrogen in minus 196 degrees Celsius. Okay, so such stored pollen grains, can be used later in our different stages. Okay, later we can be using them, and students, like how we have blood bank, we also have pollen banks. Okay, like how we have blood bank, we also have pollen banks. Clear? Yes or no? Chalo. Yes, fine. How we have this, we also have pollen banks. So we spoke about allergies, we spoke about your pollen seed bank. Okay, seed bank, and we also spoke about cryopreservation. No problem, Tanuja. Yes. Now students, we are going to go into the female. Okay, female part. So here, see, the pistil, megasporangium and the embryo sac. Okay, to now see, now the pollen grain is there, or the male gamete is present in the generative cell. Okay, generative cell. I'm extremely sorry for my cold. I did not expect it to happen, okay, when during the class. So yeah, so students, pollen grain. Okay, now what have we done so far? We have formed the male gamete. Okay, we have formed the male gamete, and that male gamete is present inside the generative cell. Okay, and where was male gamete formed? In the male gametophyte, which is the pollen grain. If this difference, if you understand, it is more than enough. Male gametophyte. Okay, so male gamete is present inside your generative cell, and it is formed inside the male gametophyte called as the pollen grain. Okay, pollen grain, because inside the pollen grain only generative cell is there. No, now this is the first part, and this is the first step of a pre-fertilization. I told you, like how marriage happened. We have found the guy. Well, okay, and the guy is completely ready. He's ready to get married. He's like, okay, I want to get married, and all of that is set, and everything like about the event, everything is set. Okay, everything is set. Now we have to make the girl ready, right? So for that we have to see the egg. So look here, to make the girl ready, we have to first understand what are the parts of it. Like how you understood about anther, here you have to understand about the pistil. So the gynoecium represents the female reproductive part of the flower. The gynoecium may consist of single pistil. Students, yes, it could be single or it could be multi. That is, what do I mean by single pistil? One stigma, style, ovary. Okay, stigma, style, ovary. It can be single, that means it can be monocarpellary. Okay, single carpel, monocarpellary. It can be multi. Yes or no? It can be multi. (3D) and all I can draw, students. Okay, so it can be multi. Multicarpellary. Got it? Multicarpellary. Now this multicarpellary can be of two categories. Multicarpellary can be fused or they can be single. Yes or no? They can decide, no, if I want to be fused or if I want to be free. So multicarpellary can be free or fused. So if they are free, we call that as apocarpus. If they are free, we call it as apocarpus, and if they are fused, we call it as syncarpus. Okay, syncarpus. So if they are fused, they are called as if they are free, they are called as apocarpus, and if they are fused, they are called as syncarpus. Okay, so in multicarpellary, students, single people have no option, only they have to stay single itself. But if they are, if they are multi people, okay, many people are there, then they can decide on being free or they can decide on going together out, right? Fused or free they can take. Free is apocarpus, fused is syncarpus. Clear? Yes. Now, see, when there are more than one pistils may be fused together, it will be called as syncarpus. Okay, students, may be free or fused. Okay, each pistil has three parts. We know stigma, style, ovary. This portion, this portion is where the pollen grain will come and fall. This is where the pollen grain will come and fall. So this is the landing place. Okay, this is called the stigma. This long structure is called the style, and this is called the ovary. Inside which, who will be there? Ovules will be there. Okay, ovules will be there. Got it, all of you? Chalo. So see, each pistil has three parts, and that is stigma, style, ovary. The stigma serves as a landing platform. Landing means now helicopter is landing. Where will it land? Right, on a helipad. So like that, a landing. Pollen grains will be scattered. Imagine Tanuja is going to play, okay, or Tanuja is going to shop. By mistake, her dress has touched on some pollen grain. Now she'll carry that pollen grain. She'll go and dust it somewhere else. So while dusting happens, this pollen grain has to make sure that it lands on the stigma of another plant. If there is not another plant, then that is gone wasted. But imagine there is another plant, it will have to land on the stigma. On the stigma, once it lands, they will talk to each other. They will have like, okay, wait, are we compatible, right? Because this is a bride and groom meeting. So they'll be like, families will be saying, okay, so stigma will check, is this fellow compatible, is this fellow fine, and then only pollen tube germination will happen. So then only pollen tube formation will happen to reach up to the egg. Is this clear to all of you? Okay, is this clear to all of you? Yes? All of you mark your attendance. Good morning, good morning. None? Okay. Now look here. The style is an elongated slender part beneath the stigma. This is basically to connect, right? Ovary to stigma, there should be a connection, so style is there. Now the basal bulged part of the pistil is ovary. Okay, the basal bulged part is called as ovary. Inside the ovary is the ovarian cavity, that is the follicle. So here where I've drawn the ovules, no? So ovules will be present inside locules. Students, this you will study in morphology of flowering plants. So I think you all have already have studied that about, you know, placental like marginations, all of you would have studied, you know, parietal, axial, etc. So that is, they are present in locules. So in, let's say in one locule you can have one ovule. Okay, like in basal placentation you will see, no, one locule will have one ovule. The placenta is located inside the ovarian cavity. Recall the definition and types of placentation. That's what I told you. Recall the types of placentation. Okay, so far good. Okay, now students, as we already discussed, we know that the stigma style ovary, or let's say carpel, okay, can be multi or mono, single or many, multi or mono. Now if it is multi, then we have apo and we have syn. So if it is syn carpel, just look at this. Okay, just look at this. This is a single, okay, ovary is there, it is placed on the thalamus, and then you have stigma and style. I mean style and stigma. Yeah, okay. Chalo. Now here what is going to happen? Here if you see, the stigma here is fused. Can you see? The stigma here is completely fused. Here, 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 they are fused. Okay. Can you all remember this? Yes, ma'am, it is fused. Understood. Okay. So multicarpellary, syncarpus, MSP and MAM. This is how you will remember. Okay. Yes, rhinovirus is not leaving me at all, and rhinovirus has become very bad person. Okay. See what? Even if I have cold, even if I have fever, I have to come and take class for you guys. Okay. So students, how do I remember the examples, ma'am? Maybe if they give this image, especially for the board students, if they give this image, then you have to remember this. So remember like this, students. M stands for multicarpellary. Okay, multi. S stands for syncarpus, and P stands for Papaver somniferum. Papaver somniferum. Papaver somniferum. Understood? Now here, ma'am, I have written, no, that is here. Can you see? All of them are individuals. Maybe the maybe the let's say in this one, what it is, stigma style ovary, that is in your carpel, you wouldn't see it, but if you see it in your anther, you will notice that maybe the filaments are attached together but the anthers are separate, right? So like that we have monoadelphous condition, diadelphous condition and all that, but as of now you do not have this. Now ma'am means what? M stands for again multi, because only multi will be able to decide if they can be free or fused. Okay, single people cannot. A is apocarpus and M is Michelia. Okay, so these are the two examples that you have to remember. Papaver somniferum you have studied in drugs chapter as well, so you can remember that. Okay, you can remember that. Got it? Yes or no? So Papaver MSP, Papaver somniferum. Okay, M for multicellular, apocarpus, Michelia. Okay, apocarpus, Michelia. Done. Now here students, here which you're seeing is an embryo sac. Okay, embryo sac. Now we have to learn about the embryo sac. Let's see first. So class 11 arising from the placenta are the microsporangia, commonly known as ovules. The number of ovules in an ovary must be 1, like in wheat, paddy and mango. Mango all of you have had, no? To many in papaya, watermelon and orchids. They're talking about the seeds. Okay, this ovules only will develop into seeds. So remember like that. Okay, yes, Papaver somniferum. Now the megasporangium, that is the ovule, let us familiarize ourselves with it, how they are formed. Okay, now students, first thing you have to know, the ovule is anatropous. Okay, anatropous. Anatropous means the funicle, the chalazal and the micropylar end is not in a straight line. Okay, not in a straight line. That is why you have anatropous. That is, funicle will go to one side, this two will come in a straight line. Now, if I have to draw this, okay, this is an ovule. In between we have the embryo sac, which I'll draw it in a different color. Okay. Sumi, it's a crime if you don't like mangoes. Seriously, I'm telling you. How can you not like such a nice fruit? So students, this is the embryo sac, right? Now these are the integuments. So look here, I've drawn a very normal one ovule because I cannot draw a lot with this pen. So look here, the ovule is a small structure attached to the placenta by the means of a stalk called funicle. This is the stalk. Okay, this is the stalk. Oh, I love mangoes. I'm a total like, I can always choose mangoes every then and now, but the problem is I'll get, this is the problem, I'll get immediately rhinovirus attack, so dangerous. Okay, so students, funicle. Now since I've drawn it this way, I think this is not you, everything went off, students. 1 second. No, it is usually we call it morning sickness, like a like once we wake up, if we have kept the fan or the AC on for the whole night, we'll get a cold. No, that's all this is. After some time it'll be fine. Okay, after some time it'll be fine. Just the early morning it will be there for 2-3 hours because of the AC and fan. Okay, so students, look here, this is the funicle or the stalk through which the ovule will be attaching to the locule. Okay, now as we go further, this which you see, those are the integuments. Okay, those are the integuments. Integuments. Okay, these are the integuments. What is the main job of integuments? What is the main job of integuments? Yeah, exactly, Tanuja. Morning cold it is. So here, students, these integuments are protective layers. Okay, protective layers. Now see here, funicle, we understood the body of the ovule fuses with funicle in a region called as hilum. This region, this region is called as hilum. The region where the funicle will attach. Okay, that is the hilum. See, thus hilum represents the junction between the ovule and the funicle. Each ovule has 1 or 2 protective envelopes. So these integuments are the protective envelopes. Integuments encircle the nucellus. Okay, now students, in between these, okay, in between these there will be nucellus. Here, this one white color which I am drawing, these are nucellus. Nucellus. Now what is this nucellus going to do? It is going to provide the nourishment for the growing embryo. Okay, it's going to provide the nourishment for the growing embryo. Now integuments encircle the nucellus except at the tip where the small opening called micropyle is organized. Okay, so the region, this is going to be the chalazal region, the opening, wherever the opening is there, that is going to be the micropylar end. Now students, you people tell me why there is the opening. All of you tell me why there is an opening. Quickly, why there is an opening? Quickly answer. Yes, why there's an opening? Answer. The opening is there because the pollen tube has to come, no? Like, see, there also sporopollen wasn't there, and that was the germ pore. Same way here also we should leave a space, no? So that the pollen tube germination can happen. So the micropylar end will have a small opening, right? Micropylar end will have a small opening that will allow the growth of the pollen tube. Got it? Yes? What happens to me? Is it clear? Okay, micropylar. A small opening called the micropylar is organized. Opposite to the micropylar end is the chalazal end, representing the basal part of the ovule. So here, this is the chalazal end. Now, enclosed within the integuments is a mass of cells called the nucellus, which I drew for all of you, so you will understand. Cells of the nucellus have abundant food reserve, okay? Abundant reserve food materials. Located in the nucellus is the embryo sac or the female gametophyte. So again, you have to learn two things here. What is that? Pollen grains is the male gametophyte. Male gametophyte, and your embryo sac. Why I'm stressing on this is many students go wrong. Okay, they will write this is the female gamete, but gamete means what? They should fuse. No, that is not happening here. Excuse me. Got it? Done. So pollen grain is the male gametophyte and embryo sac is the female gametophyte. Okay. Oh my god, it's getting worse. Okay, done. Okay, so yes, so an ovule generally has a single embryo sac formed from the megaspore. So here you can see, students, pollen grains will give rise to the male gamete. Okay, that is present inside the generative cell, and then female gamete is going to be the egg. Okay, female gamete is going to be the egg. Got it? Yes. Now, megasporogenesis. What is megasporogenesis? Megasporogenesis is the process of formation of megaspores from the megaspore mother cell. Okay, and it is called megasporogenesis. So there also we saw microspore mother cell. Here also we are going to see megaspore mother cell. MMC is going to form the megaspore mother cell. Megaspore mother cell. Okay, megaspore mother cell. Now this megaspore mother cell will give rise to the formation of what? Obviously it is going to give rise to your megaspores. Now students, here there is a catch. Okay, here there is a catch which I'll tell you here. Now look here, the MMC, that is megaspore mother cell, will undergo division and it will form megaspores. Okay, 4 megaspores. Megaspore tetrad it will form again. Okay, megaspores, 4 megaspores it will form. But the problem is, out of the 4, okay, out of the 4, the 3 will become non-functional. 3 also will become non-functional. Only this one will become functional. Okay, only one among the 4 will become functional, and that one will give rise to the egg. Okay, will give rise to the egg. So this will become the megaspore. Is this clear? Okay, so here if you see, you will be able to see the nucellus around. Okay, you can see one megaspore mother cell. That one will do form megaspore dyad, then it will form megaspore tetrad. So before the stage, okay, before the stage, there is one more where they'll form the dyad. From dyad, tetrad will be formed. Okay, from dyad, tetrad will be formed. Clear? Okay, tetrad will be formed. Now that tetrad will undergo division, and first it will form a 2-cell stage. Okay, 2-cell stage. Then it will form a 4-cell stage. Then that will only form an 8-cell stage. Okay, like this. Now this 8-cell stage will arrange itself. Okay, this 8-cell stage will arrange itself. Got it? Yes, hello. So 8-cell stage will arrange itself and it will directly form the embryo sac. So students, what is exactly happening here? What is so different from the megaspore? Have I left a space here? No. Okay, wait here. So here I'm going to write it. Okay. So here what did happen? Megaspore mother cell underwent division. She formed megaspore dyad. The dyad formed megaspore tetrad. In the tetrad, only one functional megaspore we got. Okay, one functional megaspore we've got. That one functional megaspore, okay, multiplied from 2-cell stage to 4-cell stage to 8-cell stage. So it formed a 7-nucleate 8-cell stage. 7-nucleate 8-cell stage. Now what is the 7-nucleate 8-cell stage? I will show you in the diagram. Okay, now let's go to the diagram. Here, look at this diagram, students. In this diagram, see, can you see 3 of the cells? So here 8 cells were there, no? In that 3 of them became antipodal cells, which is present on the chalazal end. So always remember CA, chartered accountant, no? So on the chalazal end, antipodal cells are there, okay? 3 antipodal cells. Now here you can see polar nuclei. This is also the central cell. The central cell area, you will have the polar nuclei, which will be considered as 1 nuclei. Okay, 1 nuclei and 2 cells. Okay, so 7-nucleate 8-cell stage. Okay, got it? Now the 3 cells downwards will have arranged themselves as synergids. Okay, they will arrange themselves as synergids and the egg. So egg is going to be present with the synergid. So this whole portion will be called as the egg apparatus. Egg apparatus. Okay, where there is going to be 2 synergids and 1 egg. Okay, now here you will see filiform apparatus. Filiform apparatus is the one that is going to help the pollen grain, telling that, hey, come inside, it will tell, no. So this pollen grain is going to come inside because of the help of filiform apparatus, because filiform apparatus is going to show the way for this fellow to come inside. Is this clear? Okay, it is going to show the way for this fellow to come inside. So look here, students. Wait, I'll start from here. Let's see what's the continuation. Okay, ovules generally differentiate a single megaspore mother cell in the micropylar region. Now we'll go continue. Okay, of the nucellus. It is a large cell containing dense cytoplasm and a prominent nucleus. The MMC, okay, the MMC is megaspore mother cell. I know microspore mother cell and megaspore mother cell is there, but they won't ask you both, okay, they'll ask you any one of it only. So here, the megaspore mother cell undergoes meiotic division. What is the importance of MMC undergoing meiosis? Okay, this is the question. Meiosis results in production of 4 megaspores. Now, female gametophyte. In majority of the flowering plants, one of the megaspores, okay, one of the megaspores is functional. I told you, no, even though 4 of them will be formed, okay, 4 of them will be formed, all the 4 will not go to the next stage. Only one will go to the next stage. only one will become functional. So one functional megaspore will be formed that develops into the female gametophyte. So here the female gametophyte is your embryo sac. Now inside the embryo sac, who is there? Egg is there. Okay, so this method of embryo sac formation from a single megaspore is termed as monosporic development. Why is it called monosporic? Because obviously only one is becoming functional, so we can call it monosporic development. Agree all of you? Right? All of you agree? Monosporic development. Okay, chalo. Now, what will be the ploidy of the cells of the nucellus MMC, the functional megaspore, and the female gametophyte, which we will see in the further part of the chapter. Okay, we will be able to see that in the further part of the chapter. Now, let's see here. See, let us study. Okay, let us study formation of embryo sac in a little more detail. Okay, now the nucleus of the functional megaspore divides mitotically to form two nuclei. Then from two nuclei it will move to a 4 nucleate stage. Okay, so look here, forming 2 nucleate embryo sac. Then 2 more sequential mitotic nuclear division will happen, that will form a 4 nucleate stage, and later a 8 nucleate stage of the embryo sac. It is of interest to note that these mitotic divisions are strictly free nuclei. Okay, that the nuclear divisions are not followed immediately by the cell wall formation. Okay, nucleus formation will not happen immediately by cell wall formation, because again you have to remember that polar nuclei is single nucleated. Okay, so after each nucleate stage, cell walls are laid down, leading to the organization of a female gametophyte or the embryo sac. So students, if I have to draw the embryo sac, we'll try as much as possible to draw with the pen. Okay, not bad, very nicely again. Now see, before this, okay, before this, let's say I'm drawing a miniature version of this. We had a 2 nucleate stage. Okay, 2 nucleate stage. Then it underwent mitotic division. Students, you have to know where mitosis is happening, where meiosis is happening. Okay, otherwise you are going to see stars in the board exam and in the questions, because you will not know where this happened. Okay, 2 and 2. Again it will undergo mitotic division. Okay, then it will go through a 8 cell stage. 1 2 3 4 5 6 7 8. Clear? Okay, clear? Yes or no, all of you? Chalo, from here, from this 8 cell stage, it has reached to a complete embryo sac. Now what is going to happen? Let's say the antipodal cells move to one pole. Okay, antipodal cells move to one pole, and that end is called as the chalazal end. Chalazal end. Now in the another end, okay, another end, there was synergids. 1 2 synergids. Okay, 2 synergids, and the most important, our heroine is here, hiding behind the security, that is going to be your egg. Okay, that is going to be the egg. Clear? So students, they have full Z-line security, that the synergids are there and the egg is there. Okay, so we can together call this entire portion as the egg apparatus. Okay, now in between this, wherever the synergids are there, there are going to be some finger like structures called as your filiform apparatus. Okay, what is it called? Filiform apparatus. Why is this filiform apparatus there? The filiform apparatus is there so that the pollen tube can aram se come inside, telling that the pollen tube, they will show the way, telling that you come this way. Okay, so pollen tube will be able to come. Now in between, okay, in the middle of this embryo sac, you have two cells which is called as the polar nuclei, and this area is called the central cell. Is the structure clear to all of you? So I will call this the antipodals. Okay, antipodals. Okay, these together I can call it as the egg apparatus. Egg apparatus. Okay, These two cells are called, okay, that is the filiform, okay, filiform apparatus, okay. These cells are called the synergids, okay. These cells are called the polar nuclei. Okay, students, this will be considered as a single cell. Okay, single cell, this will be considered as, not a separate cell. Okay, so it is going to be a, see, 1 2 3 4 5 6 7 8 8 cell, but I'm going to consider this as one, so only one nuclei I'll be considering. So 1 2 3 4 5 6 7 8 cells, 7 nucleate stage it will be, or you can write 7 nucleate 8 cell stage. Okay, now this is the female gametophyte. Now this is the place where the pollen grain has to come and fuse. Okay, pollen grain has to come and fuse. So look here, observe the distribution of cells inside the embryo sac. 6 of the 8 nuclei are surrounded by cell walls and organized into cells. The remaining 2 nuclei, called the polar nuclei, are situated in the egg apparatus in a large central cell. So this is the central cell that I've drawn. Okay, chalo. Now embryo sac is understood. Now there is a characteristic distribution of cells within the embryo sac. 3 cells are grouped together at the micropylar end. Okay, so students, this is the micropylar end. Okay, I forgot to label here. Why this is the micropylar end? Again, because the integuments are not present at that place, and also this is going to give the it is going to allow the pollen tube to come. Okay, pollen tube to come. So this is micropylar. Opposite to the micropylar will be the chalazal. Whenever you have a confusion, just think about where the integuments will be. The integuments will be there, where the filiform apparatus will be there, and where the egg apparatus will be there, right? So look here, the egg apparatus, I'm telling every line of NCERT, nothing extra I'm telling, constitute the egg apparatus. So group together at the micropylar end and constitute the egg apparatus. The egg apparatus in turn consists of 2 synergids and 1 egg cell. The synergids have a special cellular thickening at the micropylar tip called as the filiform apparatus, which plays an important role in guiding the pollen grain into the synergids. Okay, now in all of our homes, no, there will be one person who is overexcited. No, oh, someone came, we have to show them the house and all that. They'll be very excited. Like that, the filiform apparatus is. Imagine the boy is coming, the pollen tube. Okay, pollen tube is the boy. The boy is coming to the house of the female, right? So that this synergist will have filiform apparatus. She'll be like, come in, and they'll show everything. No, same way this will allow the pathway of the pollen tube. Now this which play an important role in guiding the pollen tube. 3 cells at the chalazal end are called as antipodals. The large central cell, as mentioned earlier, has 2 polar nuclei. Thus a typical angiosperm embryo sac at maturity shows 8 nucleate 7 cell stage. Is this clear? Okay, did I write ulta, students? Oh, I'm so sorry, 8 nucleate. Yeah, here actually I told ulta here. This will be considered as one cell, right? Polar nuclei, this entire thing will be considered as a central cell. So it is going to be 8 nucleate 7 cell. Okay, 8 nucleate 7 cell. Okay, I told ulta, so make the correction here also. Here I wrote 8 cell 7 nucleate. No, 8 nucleate. Okay, this is little. Okay, all of you woke up now? Soni, Tanuja, Sumi, Aditya. Okay, yeah, ulta pulta ho gaya. So ulta pulta you corrected. Okay, that is, students, this whole cell will be considered as one cell. Okay, this whole cell will be considered as one cell, so it will be called as the 7 cell. So 1 2 3 4 5 6 7 cell. Nucleus, all the nucleus will be considered 1 2 3 4 5 6 7 8. Okay, 6 7 8. So little ulta pulta happened, so just make the correction. Okay, 8 nucleate, got it? Chalo. Now pollination. So students, now see, now at what stage we are. Okay, at what stage we are? We have finished pre-fertilization. Pre-fertilization means we had to do two jobs. Okay, we had to form the male gamete. Okay, we had to get ready with the male gamete. We had to make the female gamete. Make like, you know, we have to learn about it. Now next is going to be fertilization. Okay, next is going to be fertilization. After fertilization is going to be pre-fertilization. I mean, sorry, post-fertilization. Post-fertilization. Got it? Okay. Yes. Post fertilization. So now for fertilization to happen, they have to meet, no? Okay, for fertilization to happen, they have to meet, no? So meeting will happen now. Okay, so here in the preceding sections you have learned that the male and the female gametes in flowering plants are produced in the pollen grains and the embryo sac. Okay, as both the types of gametes are non-motile, they will have to be brought together, correct? No, if you see the pollen grain also, they have to come together, right? They have to come together. If they have to come together, then because both of them are non-motile, they will not be able to come together. We need to take the help of a outsider, that is, we have to take the help of a pollinating agent. Okay, we have to take the help of a pollinating agent, okay, or a pollinator, right? Now that is going to be done by whom? No, Sumi, I should have. Okay, yes. So pollinating agent. So now look here, students. Pollination is the mechanism to achieve this objective. Transfer of pollen grains, that is, transfer of pollen grains to stigma of a pistil is termed as pollination. That means what? Now this is the okay, this is the one. Okay, now the pollen grain, okay, will be where? In the anther, right? In the anther. So inside the anther there is going to be pollen grains. Now this pollen grains, with the help of dehiscence, okay, the anther will dehiscent and the pollen grain will land on the stigma. Now students, for marriage, what will happen? Now they have to check, if the boy is nice, if they are compatible, all of that will happen. No, that will happen in the post-fertilization events. I'll teach you how they are going to look if they are compatible, and if they are not compatible, what is going to happen. Okay, if they are not compatible, what is going to happen. So flowering plants have evolved an amazing array of adaptation to achieve pollination. They make use of external agents to achieve pollination. Okay, can you list the possible external agents? That is, the external agents. Okay, external agents will be, what are the external agents you have to tell? Okay, clear? Yes or no? Clear or not? Okay, chalo. Okay, fine. Now, kinds of pollination, students, depending on the source of the pollen, pollination can be divided into three types, mainly two types. Okay, that is going to be what? Self-pollination and cross-pollination. So if I write pollination, or red is not visible, no? pollination. Okay, what is going to happen? Yes, okay, clear. It is going to be autogamy. Autogamy means auto means what? Auto mode means what? Everything will be done by itself, right? Everything will be done by itself. So we have autogamy, geitonogamy, and xenogamy. Xenogamy. Got it? Xenogamy. Very good. Now, autogamy is auto mode means what? Self mode. So I can call it simply as self-pollination. As self-pollination. Geitonogamy means what now, students? In the same plant itself, different flowers. Okay, not same flower. This one flower is there, this one flower is there. From here, anther is falling to this flower. Okay, that is geitonogamy. Okay, geitonogamy. Yes or no? Chalo. So this is the flower. Okay, this is one flower. I can draw more beautiful flower. So actually, don't you think in pen I will be able to write better? Okay, I can actually draw better. Okay, but I cannot write that much better. What do you guys think? My handwriting has become better. Okay, yes. What does Krishna Kumar Arya wants? Krishna Kumar, you can always go to you can ask your mom. Okay, she'll have better experience. Okay, so look here, students, what is going to happen? So what I've drawn looks like a bunny flower, but actually it's a very serious flower. Okay, people cannot laugh at my flower. Okay, so look here, this one. Okay, if I have to tell about geitonogamy, students, from here the pollen grains have fallen on this flower. Okay, pollen grains have fallen on this flower, so the stigma will receive this. So this is called as your geitonogamy. That means on the same plant, okay, on the same plant there were, let's say, male flowers, female flowers. Okay, so the pollen grains have fallen from the male flower to the female flower, and here also there could be one more flower. Okay, one more flower. Got it? Chalo. So this is called as geitonogamy. Now xenogamy means what? Xenogamy is fully, okay, I'm enjoying actually drawing flower. I might end up drawing the whole, one garden only here. So guys, you cannot laugh at this flower. What is wrong with this flower? You only tell me. Can you spot one difference? Okay, one mistake in the flower. So students, this is a flower. Okay, this is one flower and this is another flower. Okay, this is one flower. Now what is going to happen? The pollen from this, okay, with the help of Tanuja, Sumi, Soni, okay, other people, that will not happen, Soni. All nonsense questions you'll ask. Okay, from where your people, I don't know how your neurons work also. Okay, all crisscross questions you'll ask. This will work. This has to work. Okay, this is nature's rule. So I will make it has to work. So students, because of some pollinating agent, okay, pollinating agent like your Tanuja, Sumi, okay, pollinating agent, this pollen grains has reached. Okay, pollen grains are going to reach from the anther to the stigma of this flower, and this will be called as cross pollination. This will be called as cross pollination. Understood? Doubt, it seems. What doubt is this? I don't know. You should ask the plant. Okay, is this clear? Fine, this is cross pollination. Okay, who are the pollinating agent? It can be wind, it can be water, it can also be humans. Okay, and very importantly, insect. Okay, insect. So I've drawn, this is my students. Okay, they are the insect, like this. Okay, who wants to become this insect can become the insect. Okay, now this insect will have on its back, and it will go all the way and it will deposit here. Okay, it will go all the way and deposit here. Whoever the insect wants to be can become. So here they will become the pollinators. Understood? Such a perfect diagram. Can you be? Okay, yeah, Sony bee. Sony bee is a nice one. Okay, so Sony bee is the bee's name that we tell, and they are the most common pollinating agent. Okay, they will take the pollen grains on them. Their coat, their fur, no, their fur will have that dusty particles, so they will take that and they'll go there, they will dust it on the stigma. So that is why flowers which are attractive, like flowers which have very good color, are high chance of having proper insect pollination. Okay, ready? Understood? Chalo. Hi, autogamy. In this type, pollination is achieved within the same flower. Transfer of pollen grains from the anther to the stigma of same flower. That means what, students? This is the flower. Okay, no place only to draw my art. It looks like Tanuja is the teacher. All are asking sorry to Tanuja for coming late. Okay, now flower. See, students. Okay, this is the flower. Okay, this is the flower. Now in this flower only, obviously what is going to be there? Anther is going to be there. Okay, and stigma is going to be there. All of you have seen hibiscus plant, no? So like this, stigma also will be there. Okay, always remember, if stigma is down only, pollination will happen. Okay, if stigma, like in the sense, if anther is not above, like this is the stigma. Okay, this is the stigma. If the stigma is not above, I mean if the stigma is not below and if the anther is not above, like anther has to be like this. If the anther is not like this, then how will the pollen grains land for self-pollination? Understood? Got it? Yes. Fine. So here germination will happen. Okay, so autogamy is clear, students. So anther and stigma, complete autogamy is rather rare. Okay, autogamy is in such flowers requires synchrony in pollen. Release the stigma. Anther and the stigma should lie close to each other so that self-pollination can occur. Some plants such as Voila, Oxalis, Camellia, okay, produce two types of flowers. What are the two types of flowers? Okay, yes. Chasmogamous and Clistogamous. Okay, Chasmogamous and Clistogamous. Okay, Chasmogamous is what? Clistogamous is what? Look here. Chasmogamous flowers which are similar to the flowers of other species with exposed anther and stigma. Okay, so they will have exposed anther and stigma. Okay, exposed anther and stigma. Clistogamous means what? Okay, clistogamous means what, students? Closed flowers. These are the flowers that do not open. Okay, these are the flowers that do not open at all. So since they are not at all opening, then what is the chance for them to survive? Autogamy. Okay, their only way to survive is going to be autogamy, because they do not open. They cannot depend on another person, because if they, in case they open also, they are going to open very little, which will not allow a pollen, another pollen to come and fall. Okay, so they are not going to open, and because they are not going to open, the only option is going to be autogamy. Okay, so flowers which do not open, in such flowers the anther and stigma is lie close to each other. When anther dehiscence in the flower bed, pollen grains come in contact with the stigma to effect pollination. Thus, clistogamous flowers are invariably autogamous, as there is no chance of cross-pollination landing on the stigma. Clistogamous flowers produce a short seed set even in the absence of pollinators. Students, this one, if they don't form a short seed set, what is going to happen? Their species only will not exist, right? Because they are already closed. they do not have cross pollination, no way for cross pollination to happen, then they have to only make sure that the seed is viable, it is proper. Like imagine jasmine makes millions of seeds, okay, but the seeds are very tiny that you cannot even grow it somewhere. So in such cases, millions of seed pollinators are only the option, and that is why jasmine has such a beautiful smell, okay, fragrance, okay, fragrance, as well as the flower looks pretty enough for the insect to get attracted. Now only in these cases where they make a millions of pollen, but the pollens are very small and not very viable, not very visible, these kind of pollens will not be transferred with the help of, or will not be scattered very easily and land on a proper place. So that's why they need insects, or they need pollinators properly. Okay, is this clear to all of you? So do you think that clistogamus is advantageous or disadvantageous to the plant? Depends on the plant. No, now the plant is making assured seed set, then it is advantage only, it is not a problem for it. But the problem is there will be no variation. Okay, wherever self pollination happens, there will be no variation, right? Wherever self pollination happens, there will be no variations. Okay, yes. Now gaitonogamy, look here, gaitonogamy is transfer of pollen grains from anther to the stigma of another flower on the same plant. I already drew this and show, right? I drew this, you all remember my beautiful diagram, no? So this is one plant, this is one plant, this is one plant. Don't remember Sony's doubt. Okay, so here this is one flower, this is one flower, okay, this is one flower, okay, this is one flower. So here let's say the male flower is there, the female flower is there, then the female flower is there, the male flower might be there. Okay, so what is going to happen, students? The anther, the pollen grains from this will fall on this, okay, and this will lead to gaitonogamy. So this is not cross pollination, but the thing is it is falling on different flowers, right? From flower 1 it is falling to flower 2, so it is falling on different flowers. Okay, understood? Yes or no? So here, okay, one second, yeah. So here, although gaitonogamy is functionally cross-pollination involving a pollinating agent, genetically it is similar to autogamy since the pollen grains come from the same plant, right? They might be having slight genetic variation, but not, I mean, they are going to be genetically similar, okay? Genetically similar because it is the same flower, right? Flowers on the same plant itself. Clear or not, all of you? Okay, fine. Okay, clear? Chalo, okay. How little banana plants grow? What is the question? I think in Sumi asked. Sumi, banana is mainly grown by vegetative propagation because we can only use the stem and other parts of the plant to grow an entire banana because seeds are not viable. Have you seen the seeds in banana, right? We consume the seeds. So those are not actual seeds also, okay? So they are since they do not have actual seeds and there is no other way to cultivation, main cultivation is going to be by the help of vegetative propagation. Clear? Sumi asked, no? Yes? Is it clear, dear? Yes, okay. Tanuja asked, huh? Oh, okay, Tanuja. Oh my god. Tanuja is getting furious. Okay, yes. Tanuja, yeah. Okay, I'm telling your name and answering you. That is, banana can be planted mainly by vegetative propagation because if you have seen banana, right? Obviously, all of you have seen banana. So if you see banana like this, you will be able to see 3 spots like this, no? 3 spots like this. These are actually infertile seeds. Okay, infertile. They cannot be planted, they cannot be grown. That is why we consume it. Okay, they cannot be grown, they are infertile. Got it? Now xenogamy, students, xenogamy is cross pollination, obviously. Transfer of, what happened? Yeah, transfer of pollen grains from anther to the stigma of a different plant. This is the only one type of pollination which during pollination brings genetically different types. That means variation can be seen only in this. Okay, variation can only be seen in this. Is this clear? Variation cannot be only seen in this. Done. Okay, xenogamy. Now agents of pollination. Okay, agents of pollination, that is plants use abiotic, okay, biotic and abiotic, let's write it that way. Agents of pollination. Okay, biotic and abiotic. Biotic means what, students? Us, animals, humans, all of this. Okay, animals, humans, we are the abiotic factors. Okay, abiotic factors, again insect and all will come here. Abiotic factor, main one is wind and water. Okay, wind and water. Understood? Wind and water. Now animals, human, insect, all of them come here, but abiotic, these two people are the most important ones. Okay, these two people are the most important ones. Why they are the most important ones? Because they are going to be the main pollinators. Okay, they are going to be the main pollinators, that is wind and water, one biotic agents to achieve the pollination. Majority of the plants use biotic agents for pollination. Only a small portion of the plants use abiotic agents. Understood? Abiotic, okay, okay, abiotic agents, I told vegetative reproduction. Okay, okay, main biotic agents are going to be wind and water. Now pollen grains coming in contact with the stigma is a chance factor in both wind and water pollination. To compensate for these uncertainties, the associated loss of pollen grains, the flowers produce an enormous amount of pollen when compared to the number of ovules available. That is what they're trying to say, students. Because when the wind, it's very common, no? If wind and water is actually getting something scattered, then we have to make it in more numbers. Why we have to make it in more numbers? Because it is not assured that the wind and water, see, first of all, understand, wind and water is not your delivery guys, that they will be like, okay, I have taken the pollen from here, I have to make sure that they land here. That they will not do. So when they are carrying, many things can happen, it will get scattered in many places. So the flowers, to increase the chance of fertilization, because if fertilization does not happen, the entire flower becomes useless, right? So the flowers will make n number of, okay, n number of pollen grains. Now if you have seen hibiscus plant, okay, all of you have seen hibiscus, no? You will see that on the filament, right? This is the filament. On the filament you will see anthers. On the anthers you will see lot of powdery yellow color structures. Yes or no? Lot of powdery yellow color structures. These powdery yellow color structures are your pollens. Why they make so many? So that even while scattering, even if they scatter a lot of them also, it will make sure that they go and reach somewhere. Okay, they'll make sure that they go and reach somewhere. Clear? Okay, yes, students, will take a break and come back. Okay, after that we'll do pollination by wind, because I have to start. If I start now, I have to continue. Okay, I started the class. So you can all have your breakfast and come back. Even I'll have something and come back. It's such a nice time, no? 10:10. Okay, yeah. Hello students, am I audible? How many Tanujas in the class, yeah? Yes. Okay. So now we understood why pollination should happen, right? Why pollination should happen. Now we are going to understand if pollination is happened by wind, how is the flowers going to look like? If pollination is happening, okay? If pollination is happening, okay. Okay, the green color DP also is UD. Okay, so if pollination is happening by water, how it is going to be? If pollination is going to happen by wind, how it is going to be? See, pollination by wind is more common amongst the abiotic pollination. So they're telling that out of water and wind, the most common one is going to be wind. Students, question can come, assertion reason can come, your statement based question can come. So keep this in mind, okay? Wind pollination also requires that the pollen grains are light and non-sticky. Yeah, no, if the pollen grains are very sticky, they will not get transferred by the wind. Yes or no? So the first thing that they are telling is the pollen has to be light and non-sticky. Okay, I'm just going to write the main words here so it'll be easy for you. Light and non-sticky, okay, so that they can be transported in the wind currents. They often possess well exposed stamen. Very important. If they do not have well exposed stamen, then the wind is not going to take the pollen grains. Okay, so they have well exposed stamen. These are all general, you can just think of it. Okay, if wind has to take it, it has to be light, it has to be non-sticky, they should have an exposed stamen so it can get scattered. Okay, very normal thing. They often possess well exposed stamens so that the pollen grain can be easily dispersed, and large often feathery stigma, because students, just by having a longer stamen will not help. The stigma also should be feathery and long so that when the pollen is coming, they are able to capture it, right? They should be able to land. The pollen grains have to land on the stigma, so they will often have large, okay, large feathery, okay, large feathery stigma. This is also very important. Okay, large feathery stigma, so that airborne pollen grains can come and fall. Wind pollinated flowers often have a single ovule in each ovary. Okay, they'll usually have single ovule in each ovary. Okay, and numerous flowers packed in an inflorescence. So they usually have inflorescence, because see, the look of the flower and the size of the flower will not matter that much, because it is not insect pollination. In insect pollination, we bother about the size of the flower, we bother about the color at which what they are, all this we bother. So here it is, can be an inflorescence. A familiar example is the corn cob. So the example here given is corn cob. Okay, the tassels that you see are nothing but the stigma and the style which wave in the wind to trap the pollen grains. Wind pollination is very common in monocots. Okay, especially your grasses. Very common in monocots, especially your grasses. That is because they cannot bear very attractive flowers. They can, even if the flowers are there, they will form into an inflorescence, because individually they cannot form the flowers. Got it? Now, pollination by water. Okay, pollination by water is quite rare in flowering plants, and it is limited to 30 genera. So this is very important, students. Even though we told that abiotic pollination is very common, but water pollination is quite rare. Okay, it is only fixed to 30 genera. Only 30 genera of plants will be doing water pollination. Yes, corn cob, very nice. Mostly monocots. Okay, and water pollination will be also done mostly by monocots. Okay, monocots. Now, as against this, you would recall that the water is a regular mode of transport for the male gametes. Now, students, imagine there is a flower here. Okay, one flower is here. This flower has released its pollen grain. Okay, release the pollen grains. Now, how will the pollen grains get scattered in water? it has to go with the help of water currents. Yes or no? It has to move with the help of water currents. Now once it moves with the help of water current, it has to reach the female, female gametophyte or the egg for the fertilization. So here they're trying to say that water is a regular mode of transport for the male gametes among the lower parts of the groups such as algae, okay, bryophytes and pteridophytes, right? That is why pteridophytes is called amphibians of the plant kingdom, because they need one, at least in one stage, they need water as the medium for their reproduction to happen. So it is believed, particularly for some bryophytes and pteridophytes, that their distribution is limited because of the need of water for transport of male gametes and fertilization to happen. So what they're trying to say, they're trying to say that water pollination is very, very rare. Even if it is there, it is only present in 30 genera, okay, and mostly seen in monocots. Now the male gametes will be transported with the help of water currents, and in bryophytes, pteridophytes and algae, it is believed, or it is there, that one stage of their life cycle they will require water for the transport of the male gamete, because of which they cannot have extreme variations, right? Students, that is always going to be an hindrance, right? Hindrance compared to any other plant, that is going to be an hindrance for these plants. Now examples of water pollinated plants are going to be vallisneria, okay, vallisneria, okay, vallisneria and hydrilla, okay, hydrilla, which grow in freshwater, and several marine seagrasses such as zostera. Even zostera is an example of a seagrass, okay, sea grass. These are the examples of water pollination. Now not all aquatic plants use water for pollination. In majority of the aquatic plants such as water hyacinth and water lily, the flowers emerge above the level of the water and they are pollinated by insect or wind. So students, even though some of them are believed to be underwater, what they do is, let's imagine this is the water current, water, okay. I've become an artist, right? More than I'm explaining with the help of diagrams. So here what they do, imagine they're underwater, but during pollination what they do is they come above the water, okay, they come above the water and their flowers will be above water. Since their flowers will be above water, students, what is going to happen? Pollination can happen with the help of wind or with the help of insects. Okay, with the help of insects. Understood? So their pollination can happen with the help of insects or wind. Wind or insect. This way their chances of pollinating will be high. The percentage will be high. Okay, got it? Yeah, artist. Okay, pollination, their chances of pollination will be high. Now look here. The flowers emerge above the sea level of the water and are pollinated by insects or wind, as in most of the land plants. In vallisneria, the female flower reaches the surface of water by a long stalk, and the male flower or the pollen grains are released on the surface of water. So let's imagine this is vallisneria. Obviously, this is not how vallisneria looks like. The vallisneria looks okay like this. So I cannot draw that. Now just imagine. Okay, just imagine. Now what is going to happen, students? The male pollen grains are going to come with the help of water currents. Okay, the male pollen grains are going to come with the help of water currents and they are going to land on this vallisneria. Understood? They are going to land on this vallisneria. Got it? Land on this vallisneria. Now look here. In the female flower reaches the surface of water by a long stalk, and the male flowers or pollen grains released on the surface of the water. They are carried passively by water currents. Okay, some of them eventually reach the female flower and the stigma. In another group of water pollinated plants like your seagrass or stera, what is going to happen? Female flowers remain submerged in water and the pollen grains are released inside the water. Okay, pollen grains are released inside the water. Is this clear? So in some, they will come up telling that, okay, why to wait? We will just not take the chance. We'll just come up and we'll get pollinated by wind and water. But in some of them, what is going to happen? So the flowers will remain submerged in water and the pollen grains are released inside the water. Okay, pollen grains will be released inside the water. Is this clear? Okay, yes. Now pollen grains in many species are long, ribbon-like, and they are carried passively inside the water. Some of them reach the stigma and achieve pollination. In most of the water pollinated species, pollen grains are protected from wetting. Yeah, this is very, very important, right? If pollen grains get wet, what is going to happen? They are going to sink down. They are going to sink, or they are going to mix with something and go off. Okay, they are not going to be transporting, they're not going to do their job. So here what is happening, the pollen grains will have a proper mucilaginous covering. Okay, they'll have a proper, this looks like an omelette, but yeah, this is the mucilaginous covering. Okay, they'll have a mucilaginous covering to protect them from wetting. Okay, is this clear? Protect them from wetting. Clear, all of you? Okay, sure. Now this is what we are talking about. See students, now what is going to happen? These are the pollens. Okay, these are the pollens. So now imagine the flower has come up, okay, above the water level. Then there will be, okay, then there will be male flowers. Okay, these are the male flowers. The shape is like this. That is why it looks like a bee, but it is not a bee. Okay, it's not the Sony bee. It is actually the pollen grains. Okay, these are the pollen grains or the gamete. Okay, male gamete. This male gamete will go and fuse with the stigma here, and then the pollination will happen. This is the female flower. You can see, with the help of a long stalk, they will try to come above the flower, water, or they will come in the, maybe they will be submerged, but they will not be fully submerged. Okay, they'll be submerged, and because of it, the pollen grains will come, and the water currents will bring the pollen grains with them. Got it? Now students, is it necessary for the water pollinated flower to be colorful? Tell me yes or no. Yes or no? Tell me quickly students, is it necessary for the water pollinated flower to be very, very colorful? Yes or no question. Yes or no question. Can you guess? Think. See, for if they want to be fertilized or if they want to be pollinated by insect or wind, then they have to be colorful and a little showy. But if they are submerged, they do not have to be that colorful or showy. But the most colorful and showy flowers should be seen in insect. Okay, insect pollinated flowers. Yes or no, all of you believe in that? Insect pollinated flowers should be the one who is going to be like what it is, who is going to look more colorful, right? Who is going to look more colorful. So if it is pollinated by insect, okay, we call one more word, entomophily. Okay, entomophily. Got it? Yes. Yes. So it is no. Okay, Tanuja, it is no. Okay, it does not have to be showy until they want to, like in the sense, until they have to come above the sea level. Okay, sorry, above the water level. Students, if it is pollinated by insect, it is called entomophily. What about wind? Can anyone tell me what about wind? So, abiotic. Wind pollination, can someone tell? I taught you people on Vedantu NEET English. Water you can guess. Water you can guess. Okay, wind will be called as anemophily. Yes or no? Wind will be called as anemophily. Water will be called as hydrophily. Okay, water will be called as hydrophily. So anemophily and hydrophily. Anemophily and hydrophily. Now if it is same thing with insect, it will be called as entomophily. Entomophily. Okay, entomophily. Yes? Okay. Now here, look here. Majority of the flowering plant uses a range of animals as pollinating agents like bees, butterflies, flies, beetles, wasps, ants, moths, birds. All of this is going to help in the formation, help in pollination. Okay, all of this is going to help in pollination. Now bats are common. Okay, bats are common pollinating agents. Among the animals, insects, particularly bees, are dominant biotic pollinating agents. Bees are the most common pollinating agents. Even larger animals such as primates, okay, then tree dwellings, rodents, or reptiles, gecko, lizard, and garden lizard, all of this also has helped in pollination. Not just bees and butterflies, even the tree dwelling, the gecko lizard, even your garden lizard, all of them have to be pollinating. Yeah, even wind pollinated flowers do not have to be that showy, but they have to make sure that they have long stigma and style. Okay, stigma as well as anther. That is very necessary. Okay, now they have also been reported as pollinating species. Often flowers of animal pollinated flowers are specifically adapted for a particular species of animals. That is, here, look here, majority of the insect pollinated flowers, okay, majority of the insect pollinated flowers should be large, colorful, then only insect will get attracted. No, large, colorful, fragrance should be there. I told you jasmine example, no? Fragrance should be there. They should also be rich in nectar. Why they should be rich in nectar? Because that is the reward that they will give. Okay, for the insect for doing pollination, the reward that the flower gives for the insect is the nectar. Okay, is the nectar. So when the flowers are small, what they have to do, students? They have to form an inflorescence. They cannot have small flowers, or they have to do modifications. That day we had showed you, we had showed you about how the bract had become a modified petal. Right, it has become colorful and it was modified because the actual flower is very, very small. So if they are small, they have to make an inflorescence, or they have to make it, modifications have to come. Okay, animals are attracted by colors or fragrance. Flower pollinated by flies and beetles secrete. So here you have to remember that not every flower is beautiful, not every flower smells well. Okay, that is, the flowers which are being pollinated by flies and beetles, okay, those give out foul odors. Okay, those give out foul odors to attract these animals. Okay, to sustain animal visits, the flower have to provide reward. That's what I told you. There is a reward mechanism that, okay, you have pollinated this time, so I'm going to give you the reward as nectar and pollen grains. Okay, I'm going to give the two floral rewards that I have. First one is going to be nectar, so that the bees or the flies, butterflies will come back again. Okay, nectar. Second one, I'm going to give you the pollen grains. Now this pollen grain, students, is very simple. Okay, let's take, this is our Sony bee. Okay, this is Sony bee. I cannot draw it with black, so I'm just drawing it with green. Okay, this is Sony bee. Now this Sony bee is going to come, and what is going to happen? Let's say there is pollen grains, right? The pollen grains are going to stick to the body of the Sony bee, even to the tips of their petal, their wings. All of this, the pollen grains are going to stick. Now since the pollen grains are going to stick, so this one will go to another plant, right? It is going to take, is it going to take nectar from only one flower? No, it is going to go to another one. It will dust. Okay, it will dust these, okay, it will dust these pollen grains onto the next flower. Okay, let's say this is another flower. Okay, this will dust these pollen grains onto this flower, and if they are going to accept, okay, if they are going to accept, that is, the flower feels that, okay, they are compatible, then here there will be cross pollination happening or xenogamy happening. Okay, cross pollination or xenogamy happening. Understood? Yes, sorry, not petals, wings. Okay, teaching continuously flower, I have forgotten the parts of the animals. So here, students, from the flower the animal visit in contact. Okay, so they are going to give reward. Yes, they're going to give a reward. Don't forget that. They're going to give floral rewards. Floral rewards are going to be given. These floral rewards can be of two types. It could be the nectar, or it could be the pollen grain. Okay, it could be the pollen grain. Got it? Now, in case, okay, in case if it is pollinated by flies and beetles, then they are going to give a foul odor. Okay, foul odor. Clear, all of you? Okay. So mean, I would never do that to Sony. Okay. Yes. So students, a coating on the pollen grain, which are generally sticky to the animal pollinator flowers. When animal carry the pollen grain on its body, it comes in contact with the stigma. I showed you know how Sony bee is going to take the pollen, and that pollen the bee is going to go and dump, like, you know, what do I tell students? Like, I'm just making sounds now. Wait, they will go and. They'll come in contact with the stigma. Okay, it brings about pollination. Okay, in some species floral. Okay, in some species floral reward. So in some species the floral rewards are in providing safe places to lay eggs. Okay, safe places to lay eggs. An example is the tallest flower called as, okay, Amorphophallus. Okay, this is a tall flower. What is this going to do? It is going to help in keeping the moth egg. Okay, moth egg inside them. Okay, at the same time you have a species of yucca plant. Okay, yucca plant and moth. Okay, Pranuba moth. Okay, yucca plant and moth, they also have the same, like, you know, symbiotic relationship. Like, they'll help each other. Okay, what they're going to do, students, the moth will deposit its egg in the locule of the ovary. So here the yucca plant, okay. Okay, so the yucca plant, I'm just checking if there is this plant. This is the Amorphophallus flower. Okay, so students, this yucca plant plus the Pranuba moth. Okay, Pranuba moth will, what they are going to do, this Pranuba moth will lay the ovaries. Okay, the eggs in the yucca moth's locule. Okay, locule of the ovary. So this is like a safe place where they can keep this. Okay, safe place where they can keep the egg safe, and this yucca moth also will help. Okay, now the larvae of the moth will come out of the eggs as the seeds start developing. Understood? Understood? Yes or no? Yes or no? Yes, symbiotic relationship. Okay, they show a symbiotic relationship. Symbiotic relationship. Even if it's not symbiotic, no students, even one person is getting benefited but then other is not getting harmed, like a commensalism, we can say. Symbiotic, both should be benefited. Here maybe only one is getting benefited because the moth is able to you know, put the ovary or the egg in the ovary, and it is going to be a safe place for the moth to develop, right? It is going to provide a shelter. Okay, same thing goes with ants. Okay, is this clear? Okay. Now, why don't you observe some flowers of the following plants? Students, your job it is to observe the flowers of your following plants. If you don't observe, no, then this is going to be a problem because NCERT is telling. So you can read this. They're just trying to asking you how the flowers will look, what will happen if they come in contact with the animals. All that is not needed. But this Yucca and Pranuba moth you cannot forget. Okay, Yucca and Pranuba moth you cannot forget. At the same time, you cannot forget Amorphophallus plant also. It is a 6 feet plant. Okay, taller than both all of us. Okay, it's a 6 feet plant, and inside this also the insects will settle or add their seeds. Okay, I mean, what am I telling students? Add the eggs. Okay, add the eggs. Yes. Now here, outbreeding devices. Now what is outbreeding devices, ma'am? You just tell us generally. See, I'll tell you. Oh, where's, I did not leave a slide. I don't know how come I did not leave a slide. Okay, students, outbreeding devices. Before I tell you anything about outbreeding, let me tell you about inbreeding. Okay, inbreeding. There is a depression called as inbreeding depression. Students, plants also go through depression. Not only you, plants also go through depression. Okay, inbreeding depression. That means continuous. Okay, continuous self-pollination. Continuous self-pollination leads to inbreeding depression. Clear to all of you? Continuous self-pollination, right? Okay, continuous self-pollination will lead to inbreeding depression. That is, they are not able to have any variations. Because they are not able to have any variation, they will go through inbreeding depression. Okay, now it is plant's responsibility to protect and make sure that the plant is going to survive for a longer time. If inbreeding depression happens, it is going to lead to no variation, and it is also on a long run going to damage the plant. So the plant will take some techniques to do something called as to avoid inbreeding depression, and that technique is called as outbreeding devices. That technique is called as what? Outbreeding devices. Clear? Outbreeding devices. Now look here. Majority of the flowering plants produce hermaphrodite flowers, and pollen grains are likely to come in contact with the stigma of the same flower. So the first inbreeding device that the plant will take is, they will be like, wait a minute, I am no more going to form a dioecious plant, I will form a monoecious plant. That means I will only have one, okay, one, I will neither have androecium or I will have gynoecium. I will not have both. Yes or no? That is, they will form unisexual flowers. They will form unisexual flowers. Next thing to avoid self-pollination is like, they will be like, change the position. Okay, change the position. Change the position. That is, if this is how in plant, let's say this is scenario 1. Okay, when inbreeding depression was happening, this is scenario 1. Okay, I am drawing the scenario. Okay, 1 scenario, 2 scenario. Yes. Now the second scenario that the plant will decide to do. Okay, plant will decide to do is that plant will this is the stigma. Students, diagram is very okay, going all the sides. This is the anther. So what the plant will decide to do, plant will be like, I will grow or I will make the stigma, the height of the stigma longer than the anther. Now in this situation the anther was easily able to land on the stigma, but in this situation can they? Okay, in this situation can they? It will not, right? Because the anther is down and the stigma is above, right? So when the stigma is above and the anther is here, how will the anther be able to scatter the pollen grains? Even with the help of wind, it will not be able to possibly do it. So the second thing that the plant is going to do is going to change the position of the stigma and the anther. Okay, going to change the position of the stigma and anther. So first step that they did was to form unisexual flower. Second step is to change the position. Now look here, continued self-pollination results in inbreeding depression. Okay, this is the thing I told you. Flowering plants have developed many devices to discourage self-pollination and to encourage cross-pollination. Okay, in some species, pollen release and stigma receptivity are not synchronized. This is the third point. That is where the pollen grains will get matured, but the stigma will not be ready to take the pollen grains. Okay, that means stigma's maturity and pollen grains maturity will not be in sync with each other. Okay, they will not be synchronized. If they are not synchronized, you tell me, how will pollen tube germination happen? Okay, how will all the formation happens? Yes or no? What is happening, students? Rana, Tigerina and Americana is having a conversation. Huh? Yes or no? People are having conversation. Is it? Is it? One second, students. Just give me 1 second. Okay. So here, what is going to happen? Here, either the pollen grains are released before the stigma and becomes receptive, or the stigma becomes receptive much before the release of pollen grain. That means what, students? Both of them will not mature at the same time. Both of them will start maturing at the different time, and this will lead to problems. Okay, this will lead to problems. Now, neither the pollen grain is released. Okay, this is done. Okay, so here receptivity and synchronization changes. Okay, in some other species, the anther and the stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. Both these species, okay, both these devices prevent autogamy. The third device to prevent inbreeding is self-incompatibility. That is, the stigma will be like, wait a minute, I am not going to become, like, in the sense, I'm not going to accept my own pollen grains. I'm going to make sure that it becomes, like, compatibility will not be there. Okay, compatibility will not be there. Understood? This is a genetic mechanism and prevents self-pollen from same flowers or other flowers of the same plants from fertilizing the ovules by inhibiting pollen germination or pollen tube growth. So what they will do, they are going to, yeah, it's my alarm. Okay, so what they are going to do, students, they are going to make sure that I am not compatible, right? So when self pollen comes and falls, they'll be like, wait, you are not compatible. I'm going to reject you and I will not let you grow the pollen tube on the stigma. So the stigma will decide like this. Okay, so around 4 points are there for inbreeding depression. So you can remember and keep: unisexual flowers, change in position, synchronization will be changed, and also self incompatibility. Okay, this is a genetic mechanism and prevents self pollen from the same flowers or the other flowers of the same plant from fertilizing the ovules by inhibiting pollen germination or pollen tube growth. Another device to prevent self-pollination is the production of unisexual flowers. If both male and female flowers are present on the same plant, such as castor and maize, it prevents autogamy but not geitonogamy, right? Geitonogamy cannot be prevented. For that, you will have to do artificial hybridization, like you have to do emasculation, you have to do bagging, etc. Correct? No? Yes, students? Clear? All of you? Why aren't you guys marking the attendance? Okay, why aren't you guys marking the attendance? Tell me. Tell quickly. One second. What is happening? Okay, ready, all of you? Chalo, correct, clear. Okay, chalo. Now pollen pistil interaction. See, one of the steps that this stigma told was self-incompatibility. No? Now what is going to happen, students? Now this is the stigma. This is the stigma. So the moment the pollen grains come and fall on the stigma, different pollen grains. Remember, I had taught you people this in Vedantu NEET English with the example of marriage. How many of you remember? Students, this is the 3 different pollen. Okay, now what is going to happen? How do the stigma know which pollen I am supposed to allow for pollen tube germination? So first ever step that they're going to do is something called as chemical interaction or chemical dialogues. Chemical interaction or chemical dialogues. That is, they are going to talk with each other. Let's say one is a doctor boy, one is an engineer boy, one is a bank manager. The girl is going to talk, no, telling that, okay, what are you going to do? Then he will tell, I will take you to abroad trip. I have a lot of money. I will make sure that your health is proper. So they'll keep having dialogues or chemical interactions. So first chemical interactions will happen. After chemical interactions, the stigma will understand with which they are compatible. If they get to know that I am compatible, they will allow the pollen tube formation. Okay, they'll allow the pollen tube formation, and then the pollen tube will extend all the way up to the ovary, right? So let's say it will extend all the way up to the ovary. Okay, 1 second, students. It will extend all the way up to the ovary, and in the embryo sac, who is going to be sitting? Filiform apparatus will be sitting, right? This filiform apparatus will tell the pollen tube that, okay, you come inside, and this way you will be able to enter inside the egg. Understood? Enter inside the egg. Yes or no? Clear? Clear or not, all of you? Okay, chalo. So the compatibility depends, right? So what is going to happen, students? The pollination does not guarantee the transfer of right type of pollen. I told you they are not delivery people. They will not look at the location and be like, okay, this pollen has to fall in this stigma. Whichever pollen is coming by wind, water, by insects, by humans, it will come and fall on the stigma, because stigma has a sticky substance. Pollen also has a sticky substance, so they will come and fall. Okay. Now once they fall, they will check the compatibility, if the right pollen has come and landed or not. Okay, now the pistil has the ability to recognize the pollen, whether it is the right type or the wrong type. If it is the right type, the pistil accepts the pollen and promotes post-pollination events. Okay, post-pollination events, that is, if the stigma feels that, okay, the right pollen has come and fallen on me, then they are going to continue. Okay, they are going to continue the pollination to happen. That is, they are going to be like, okay, you are compatible to me, I will let you grow. Okay, compatible to me, I will let you grow the pollen tube. I will let you grow the pollen tube and extend all the way up to the egg. Got it? Extend all the way up to the egg. Is this clear? Okay, so here you can see the image of it, students. You can see the image of it. This is the wrong pollen, this is the wrong pollen. The stigma did not allow the pollen tube germination, but you can see this one, pollen tube germination has happened. It is going all the way. So inside this, what is going to be there? Here generative cell is going to be there, no? The male gamete is going to pass all the way and it is going to fuse with the egg cell. Okay, and here you can see how they are coming. See, male gametes are there, vegetative cell nucleus is there. Male gamete will enter through the filiform apparatus and go and fuse with the egg. Okay, go and fuse with the egg. Is this clear? Whom are you guys telling good morning to? Yes or no? Is this clear, students? I think my students started imagining that they're seeing Diksha ma'am and Baswa sir every time. Okay, so this tubes have entered inside, right? Tubes have entered inside. I cannot see Baswa sir. Where is Baswa sir? Oh, clear. Okay, I can see. Okay, so the tube will enter inside and they are going to do the fusing. So the male gamete plus the egg is going to fuse. Okay, and this is going to continue. Now, this is going to continue. Now I can see. So students, here it can be at a 2 generative cell stage or it can be at a 1 generative cell stage. Okay, this 1 generative cell is the actual groom who came. Okay, he is the first boy who came to see this girl. This fellow, one more comes to, that is going to be the fake fellow. So that fellow will go and combine with the polar nuclei, and this one will go and combine with the egg. Okay, so this will give rise to endosperm, this will give rise to your zygote or the embryo. Clear or not? Clear or not? Okay, students, I'm going to stop here because I have a very important task at 11 o'clock. Okay, I have an important meeting at 11 o'clock, so I will not be able to continue the class, so I'm stopping right here. So yes, thank you so much for joining. See you all in the next class, and the next class will be very soon. Tomorrow morning itself I'll take the next class, and tomorrow we'll try to finish as much as possible. I think we'll finish the chapter itself because we have done with pollen pistil interaction. Now I'll teach you about how double fertilization happens, triple fusion happens. Okay, by this portion will be over, sexual reproduction in flowering plants. So see you all tomorrow with another portion of your sexual reproduction and flowering plants. Do not forget to mark your attendance. All of you hit that like button. Nothing is going to happen if you hit the like button. Okay, it is actually one way of showing that ma'am, we liked your class or we understood something from the class. Okay, so yes, see you all tomorrow, students. Bye bye, take care, and yes.